Design.
So I would like to welcome everyone to today's webinar. It's 5:30 p.m. in Europe and 11:30 a.m. on the East Coast of the US. We're getting started on time. Today's webinar is entitled Pharmaco Imaging, Potential in Translational Research, a case study of EGFR PET radiotracer, which is being presented by Philippe Sloss for the corporate introduction and Cyril Berthet for the rest of the presentation. Before we start, I would like to introduce myself. My name is Lucie Perrault. I'm a Business Operation and Marketing Officer at Oncodesign, and I will be your host for this webinar. First, I'd like to address a few practical matters. This webinar will be available to all attendees. We will send you a link to this presentation by email. Directly after the presentation, our speaker, Cyril Berthet, will answer a few of your questions. So if you have any, please type them in the control panel that you can see on your screen. The other attendees won't be able to see your questions if you send them directly to Cyril or to the host. If you cannot see the control panel, just click on the top arrow on the top of your screen. In case there are questions that cannot be answered by the end of today's session, Cyril will respond to you by email within 48 hours. If you are experiencing technical difficulties, please send a message to the host and someone will be able to assist you. I hope that you will enjoy this presentation. So now, let me introduce Philippe Sloss and Cyril Berthet, our speakers of today. Philippe holds a PhD from the Louis Pasteur University of Strasbourg in France in molecular biology and bacteriology of lactic acid bacteria. He moved to the field of oncology in 1996 with a focus on immuno-oncology with the setup of murine models to support the clinical development of gene therapy vectors encoding cytokines as a strategy of non-specific immunotherapy of cancer. He joined Oncodesign at the beginning of 2016 as Director of Scientific Operations, interacting closely with Oncodesign clients to support their evaluation of candidate therapies through preclinical studies. Cyril Berthet, he is a Discovery Program Director at Oncodesign. He holds a PhD in molecular and cellular biology from the University of Lyon in France. In 2002, he joined the mouse cancer genetics program at the National Cancer Institute in Frederick in the US as a research associate. Cyril joined Oncodesign in 2007 as a project leader, and he now manages strategic partnerships in therapeutic and biomarker discovery. He is particularly involved in Pharmaco imaging R&D programs and leads the PharmImage consortium. So having said that, let's begin the presentation. Philippe and Cyril will guide you through the slides that they will show you today.
So thank you, Lucie, for the introduction. So a few slides for the introduction to Oncodesign. So Oncodesign is a biotechnology company that's aiming to bring therapeutic solutions to patients with no current treatment options in oncology and other major diseases. So as you know, we have a compound, we have entered the clinic, and Cyril will come to that. And so also we have a strong pipeline in multi-kinase microscopic inhibitor at different stages. So historically, is a classical preclinical CRO which encompasses more than 150 employees. So the majority being in Dijon, the historical site. So we have also a site in Montreal in Canada, and so recently a research center close to Paris was acquired in the 1st of December. So the company was created in 1995. The revenue of the company were close to 15 million euros in 2015. The majority of the revenues are generated outside of France, and 40% of the incomes are invested in research and development. So so the site in Dijon is close to 3,000 square meters. It's a little bit smaller in Montreal, and so as I was saying previously, a big research center was acquired close to Paris, close to more than 10,000 square meters. So we have major and big clients like Bristol Myers Squibb, Boehringer Ingelheim, Gilead, Ipsen, and Amgen. So a major recent acquisition was from the GSK research and development side. It was founded in 1987, so in the suburb of Paris, Les Ulis, within the Paris-Saclay research cluster cluster. It's a state-of-the-art facility that was rebuilt in 2010 that include highly experienced scientific staff in medical chemistry, biology, pharmacology, and DMPK. This acquisition meets all points to Oncodesign strategic approach, and it will accelerate our growth in the market of therapeutic innovation by scientific excellence. So it's complementary to the classical oncology tools of Oncodesign. It will allow the enrichment of the pipeline for all our projects, and it will extend the experience and, most important, additional capabilities in the development of all drug discovery projects at the service of strategic collaboration and partnership. And it will allow us to constitute an integrated and drug delivery services offer beyond oncology. So historically, there are two major assets in the company: the experimentation, so more than 20 years old now. So this part has tested more than 3,000 molecules throughout the history. There are on average more than 250 preclinical studies per year in that division, and we have more than 500 clients that includes 14 big pharma of the groups. The site is accredited. The discovery part of the company is more recent. It has been created in 2008. It has created a lot of high potential business. So from a strict research point of view, a lot of patents were delivered, more than 16. And we could, during these years, could have some lucrative partnership with Bristol Myers Squibb, Ipsen, or UCB in Belgium. So Oncodesign has a long-standing experimentation knowledge and know-how supporting the high potential discovery business. And the recent acquisition of GSK Research and Development Center and scientific staff with strong discovery track record. In terms of technology platform, so there are several ones. If we begin from the top on the right, we have the TKIs with humanized animal models and patient-derived xenografts. So concerning the Pharmaco imaging, so it's a very important asset, and I won't go too long because Cyril will talk about that. The Predict platform is the classical PK/PD pharmacology of the company with mouse model and rat models. A recent platform is TOT, time on target, that will encompass cell-based tests to predict and speed up the progress of Oncodesign research program. An important asset is also Nanocyclix, so with more than 7,500 medical chemistry macrocyclic kinase inhibitors, which are currently in high investigation. And the most recent platform is Oncodesign, that includes bioinformatics modules to identify those patients that are resistant to available therapies to characterize them. So now I give the floor up to Cyril for the rest of the presentation.
Thank you, Philippe. So now we'll focus on our program on PET tracer. As Philippe just mentioned, this is part of our platform PharmImage, dedicated to all treatment and biomarker follow-up through Pharmaco imaging. We have different platform in Pharmaco imaging, chemistry probe toolbox, Compound radio labeling, we are able to label small molecules, as I will show you, and up to antibodies through chelation and organometallic chemistry. We have access to all equipment, imaging equipment, PET, MRI, SPECT, and luminescence, and we are highly involved in a preclinical prototype for PET MRI. And also we are able to to do first-in-man administration of PET radiotracer through our partnership with a clinical center. So all these platforms go into our Pharmaco imaging assets, and we can truly have a continuum between chemistry to clinics. So the IMAkinib project is an innovative approach based on Nanocyclix Tyrosine Kinase Inhibitors, as Philippe just described, which aims to develop new radiotracer for PET imaging. TKI PET-imaging can provide diagnostic tools to determine and predict the activity of kinases and the responsiveness to TKIs. In this program, we are looking for different targets. I will show you today our project that has been developed on EGFR, the epidermal growth factor receptor, which is known as an established target for the treatment of advanced non-small lung cancer. As you know, TKI targeting EGFR, such as gefitinib or erlotinib, are standard treatment of tumors harboring EGFR mutations. In example, L858R, which is the most common one. But unfortunately, the majority of patients develop a resistance to the TKI within 1 year. And most of them, this resistance is related to a secondary mutation, T790M, in EGFR, which is in the gatekeeper of the kinase. And our programs, the goal of our programs really was to answer clinical needs about the diagnosis and the follow-up of the responsiveness of the TKI, EGFR TKI, by using a specific EGFR targeted PET radiotracer that we could use in clinic for diagnosis and treatment follow-up. So here is a flowchart of our radiotracer development, starting from our library of highly potent and selective Nanocyclix kinase inhibitors. We are looking for targets of clinical interest, selected on overexpression or activity in tumors. Clearly, we are focusing on kinases so far, and we go very quickly to radiochemistry. We do a radio labeling with fluorine, and the characterization in vitro and in vivo. The in vitro is mainly done by binding assessments in biological samples, cell line first, and tumor, patient tumor very quickly. And we do the biodistribution in vivo to evaluate the ability of our compounds to hit the target, and in our case, the tumor. Based on this proof of concept, we prepare all the regulatory files and the toxicology to do the first-in-man administration, which is a phase 0, phase 1 clinical trial. When it's done, we can move to phase 2. With our current program, all this flowchart has been done, and we are currently in phase 0, phase 1. So the challenge of PET tracer development is quite different than for a therapeutic program. The properties of the compound are very different, and we have a specific target product profile based on the needs of the PET tracers. First of all, we identify a clinical needs where PET tracer may bring complementary benefits and not compete with omics technology. The cost of PET tracer development is quite high, and willing to compete with other diagnostic tools that could be based on blood sampling will be a waste of money and time. So we clearly identify a clinical needs where PET tracer can bring some new assets. So of course the biodistribution studies and labeled drug, which is the first thing that we can do when we work in parallel of a therapeutic program, but we want to go beyond that with an application in companion diagnostic for the selection of patients, but mostly for a non-invasive treatment follow-up to be able to predict the efficacy and the identification of resistance, which we think is one of the key benefits of a tracer that can bring to the patients. And of course, the localization of metastasis with regard of the targeted molecular profile is something that is unique to the PET imaging. Then we select very carefully our compounds with very specific profile for PET tracer development. The key point is, of course, the high affinity and the selectivity. Slow binding release in vivo is, of course, important, at least within the half-life of the fluor 18. The feasibility of the fluorination, the radiochemistry is also key. We need to add the fluor at the last step of chemistry, and so this is something that needs to be looked up front. And for the pharmacological profile, what we basically need is something that is compatible with IV injection and image acquisition. We need to have a specific binding that led us to have a clear difference between in vivo background versus specific target binding. We think that our Nanocyclix library provides advanced targeting compounds that are valuable for PET tracer development. And one of the points is that the radiochemistry is favored through the use of standard fluorination strategy on the linker that we have on our microcycle. So a bit of background about the EGFR mutation. So EGFR mutation is seen in 15% of non-small cell lung cancer. This is a high amount of NSCLC. And currently, the treatments that are given to the patient are given only if the patient carries the mutation, one of the activated mutation of EGFR. This is a clear criteria for patients selection and EGFR targeted therapy. ERSR and TAR-SEVA, and now there are some new ones are coming, are the most well-known TKI treatment. EGFR expression itself is not well correlated with clinical patient outcome, and this is really the activity that is important, and the activity is correlated actually to the mutation so far. As I say in the introduction of the talk, unfortunately, the majority of patients will develop a resistance to the TKI within 1 year. That is related to a secondary mutation in the gatekeeper of EGFR, the mutation T790M. And when this mutation is acquired, the TKI, the therapeutic TKI, such as the ERSR or TAR-SEVA, do not bind anymore to EGFR, which leads to the growth of the tumor. So when we address the clinical needs, we consider that the EGFR radiotracer must be a marker of the activity of the kinase EGFR. That is, of course, correlated with its mutational status. And then the EGFR activity under treatment should be monitored by the radiotracer to propose alternative therapies to the patients when the secondary mutation is acquired and when they are resistant to the tumor. The tumor are resistant. So here, just a small cartoon to illustrate our hypothesis for diagnostic application of a tracer. So at diagnosis, there is always a biopsy, and now in most of the worldwide, everyone is looking for the mutation of EGFR and decide on the treatment with the TKI depending on the EGFR status. We could do a PET scan here, but there is no high benefit commercially compared to the biopsy and to looking on the mutation. But on the other hand, we can look at metastasis and see if there is difference in metastatic status of EGFR. The key point is to look very quickly at the efficacy of the treatment. So after 1 week, we can do a second PET scan and see if there is a signal decrease that should indicate a response to anti-EGFR treatment by competition between the treatment and the tracer. And after a few months, we can monitor the EGFR response and the resistance when there is reactivation of EGFR, even upon treatment when the secondary mutation occurs. The EGFR activity will come back, and we should be able to monitor if the tracer bind also to the secondary mutation. So just to highlight how we built our program, this is the target product profile for our radiotracer. So at first, of course, we define the area, the clinical needs. Here is the old EGFR-driven mutation, and at first we focus on the lung cancer, the NSCLC lung cancer. The target population is the patients that are cancer known to be driven by EGFR. We are looking for compounds when administered in vivo, the target affinity. So the injected dose, the percentage injected dose per gram should be above 1%, at least ideally, and above 0.2% is good enough to see a signal of what it could be depending on the noise. The tumor muscle ratio in that case should be above 4. It's a good ratio, and above 2 is acceptable. The target selectivity is key. We are looking for 100-fold between the EGFR mutants, the activated EGFR, versus the wild-type EGFR and other targets. The mode of administration is IV. It's only one administration. The safety tolerability, with the low amount of compound that we administer to the patient, it's mainly the adverse events that are related to the fluor radioactivity, which is well described. Radiolysis is important, should be below 15% ideally. Formulation is usually not a major issue. The compounds with the low concentration that we administer is usually good in physiologic serum. Stability of the precursor is important for industrial purposes, if possible above 2 years, and if we can have half-life that is much higher than the fluorine is of course important. Radiosynthesis should be lower than 1 hour with only one step ideally, and the purity, radiopharmaceutical purity should be above 99% with a specific activity above 50 gigabecquerel per micromolar. In human, the injected dose that we calculate should be above 1.4 micrograms. So this is the kind of template that we use for target product profile to really drive our program and reach our goals to have a PET tracer that is usable in clinic and not only as a preclinical tool. So our compounds, which is named ODS2004436, that we call 4436 now on. This is a table that summarizes the in vitro properties. The biochemical IC50 is in the nanomolar range, around 5 nanomolar on EGFR wild type and mutated, and 500 nanomolar in the double mutation when the secondary with the T790M. Here we have a high level of activity on the wild type, but this is a biochemical assay, meaning that in this setting EGFR is activated. This is why we have a good activity as well. More or less, we are in the same range than gefitinib, except on the double mutant where we are more active. The cellular IC50, as you can see, there is 3 cell lines, which of them match with the EGFR wild type, single mutation or double mutation. And as you can see on the wild type, the H4041 cell line, we are losing activity with our compounds. We are above 42 micromolar. So when EGF is not activated, we do not observe any activity. In the cell line 3255, with one mutation of EGFR, we are in the 5 nanomolar range, more or less at the same level than gefitinib. And in the third cell line, the 1975, with the double mutation, we are around 5 micromolar, which is at least 3 times better than gefitinib. I will just show you some Western blot for the autophosphorylation, so I won't describe the next results, but we are more or less in the same range. And for the ADME results, the kinetic stability is quite good. The radiochemical stability and plasmatic stability are superior to 20 hours and 4 hours respectively. And for the clearance in human macrosomal stability, we are 7.7 microliter per minute per milligram of protein, which is good, and the half time is also above, is at 179, which is good. For the rats, it's a bit lower, but it's still it's still good for preclinical studies. On the right, there is a panel that represents the kinase selectivity. It's a dendrogram of the whole kinome. We have tested 92 kinases at 100 nanomolar, and only 2 kinases were inhibited, EGFR and RIP2. So we consider here that we have a good selectivity. RIP2 is mainly related to microbia signaling. So here I show you the autophosphorylation of EGFR on the free cell line with gefitinib and our compounds. You can see on the first panel on the right, on the top right, on the cell line with 1 mutation, we observe a good inhibition of EGFR as low as 10 nanomolar with our compounds and at 100 nanomolar with gefitinib. On the wild type, with NCI H4041, we observe at 1 micromolar and 10 micromolar for gefitinib, and at 1 micromolar with our compound, we start to see a signal of inhibition of the EGFR phosphorylation. Here it's in presence of EGF, so EGFR is activated. And for the third cell line, 1975, with the double mutation, we observe again an inhibition of EGF in the range of 10 micromolar for our compounds, but we do not observe any inhibition with gefitinib. We determine the KD of our compound and the specific binding by competition with excess of the cold compounds, so labeled with 19 fluor instead of 18 fluor, and with increased dose of the cold compounds. You can see here in the control you have the binding. So this is a slide of a tumor taken from after a xenograph, and this is the tumor NCI H3255. So the control is the total binding, and then you have the competition with the cold compound, and with increased activity, you can see that we are competing with our radiotracer, showing the specific binding. So the KD here was estimated at 50 nanomolar. When we do the same exercise with an excess of gefitinib, so here you have the NCI 3255 in the middle panel, we observe again an inhibition of our compounds. The binding inhibition by gefitinib is estimated around 50–60%. There is also an inhibition at a lower level on NCI H41 with gefitinib. And if we look at MCF7, which is a cell line that expresses very low level of EGFR, we do not observe a clear binding, which also is a good result to illustrate the specific binding of our radiotracer. Then we move to the in vivo proof of concept of EGFR targeted PET tracer. We did a biodistribution of our compounds, show a strong imaging contrast in tumor compared to other tissues. Here the image, on the left part of the image you have the wild type EGFR H41 cell line, and on the right part you have the cell line H3255 that harbor the activated EGFR mutation L858R. And clearly you can see that we observe a signal only on the activated EGFR cell line and not on the wild type one. So this is a clear indication that our tracer is specific to activated EGFR. And we are able to discriminate the lung tumors between resistance, so the wild type EGFR, and the sensitive mutated EGFR to gefitinib. On the right panel, you have an image of the whole body, and you can see that we have elimination of the tracer through the liver. Then we did a second experiment with another model. So on the rats, again, you have the two tumors that are engrafted on the rats, the wild type one and the single mutant one. And on the right panel, we put two models as well. Again, the mutation EGFR A48 on the right, and on the left, the EGFR double mutation A48 and T790M. And you can see the uptake in the single mutation cell line is quite high in both cases, and we do observe an uptake in H1975 models. However, it's at a lower level, which was expected since the binding and the IC50 of our compounds was lower. But we think that probably we should be able to discriminate the three types of tumor. Here, of course, it's on cell line, and now we have to look in human. We are looking in human to see if we can discriminate this type of tumors in human. We look for correlation between the phospho-EGFR expression. As I mentioned earlier on, the expression itself of EGFR is not a good biomarker for the sensitivity of the treatments, but the activity of EGFR is a good one. Looking for phospho-EGFR in clinical setting is not very good. This is why using a PET tracer that is correlated with the activity of EGFR really bring a new benefit. Here you have two pictures. The phospho-EGFR, so it's immunochemistry on the wild type cell line that has a low staining since EGFR is not highly activated, and on the 3255 with the mutation of EGFR, we have a high activity of EGFR. And if we correlate the immunostaining of each of the tumor with the uptake of the tracer in the same tumor, there is no correlation in the wild type tumor. In the middle panel, you have the correlation with the 3255, and on the right panel with the 1975 models. In both cases, we have a correlation between the uptake of the radiotracer in the tumor and the phospho-EGFR expression. This correlation is good in models that are below 1,000 cubic millimeter, and this is not good anymore with big tumors. Probably it's due to a high level of necrosis in these preclinical models, and we have to define more carefully the region of interest, which was not done as a primary analysis. For the in vivo biodistribution, so we have a percentage ID per gram, which is around 0.2% for the NCI 3255 cell line, which is good enough to have a signal, as you have seen before. And we have also an uptake in other models. If you look at other organs, kidney and livers are the two organs where there is elimination of the tracer. If you compare between 90 minutes and 180 minutes, you observe a decrease in kidney and livers, but the uptake in the tumor remains stable, which is very important if you want to be able to do an imaging over time and to increase the contrast between the tumor and the other organs. If we look at the tumor over muscle ratio, so here it's at 90 minutes. In the first part of the graph, you have the uptake of each of the models with no competition. This is around 2 for H45-1, and mostly it was below. And after 90 minutes, it was after 90 minutes. If you go to 180 minutes, the tumor over ratio decreased for this tumor, which was not the case for the third tumor, the 1975, where the tumor over ratio remained constant over 2. and for the tumor that express the high activity of EGFR, the 3255, we have tumor over muscle ratio that can reach 6 and in average around 4. If we do the competition with gefitinib, we observe a clear competition on the 3255, which was expected because EGFR is binding to the single mutant L858 mutant, EGFR mutant, and there is no competition on the double mutation, which was expected since the gefitinib do not bind the double EGFR mutant. If we perform the same competition with our cold compound, we observe inhibition, of course, on the 3255 models, but also on the 1975 models, since our compound is binding to both mutants. But these results illustrate the specificity of our EGFR tracer. And then we just did an extended single dose toxicity, preclinical toxicity studies with unique administration on 30 animals. We use rats and 30 controls. We did a follow-up for 15 days with an injected dose that was estimated to be superior to 100 Hz, the injected dose to human. Here there's a table to compare the human and rat volume, administration volume, and the dose that of the compounds, the comparative dose between human and rats. So based on the results that we do not observe any toxicity, we design our clinical trials. So this is a phase 0, phase 1. The patients all the patients with EGFR mutation that receive gefitinib or any other EGFR TKI with a continuous treatment for 28 days. The scheme of care for patients with EGFR mutation was as the following. After the biopsy and the diagnosis of the mutation, we do a first scan to see if we have a signal within the tumor, and a second scan after 8 days upon treatment to see if we can monitor the decrease of the signal after treatment. There is a second arm with wild-type EGFR patients with only one PET scan where we should observe a decrease. We should not observe any signal since EGFR is not mutated. So to conclude all these results, within the IMAkinib program, we have designed a new TKI PET radiotracer targeting EGFR that is based on our Nanocyclix technology. Other programs are ongoing, and we have now a whole flowchart that is available for internal program, but also for partnerships to move forward a radiotracer from radiochemistry to clinical PET-imaging. And everything can be performed in Pharmimage platforms. In vitro, our compounds show a biochemical profile that is comparable to gefitinib on wild-type EGFR, on L858 mutated EGFR, whereas improved activity is observed on the double EGFR mutants. In vivo studies suggested that our radiotracer 4436 binds selectively to activated EGFR and is a good candidate to evaluate the EGFR activity in lung cancer. Currently, we have initiated a clinical evaluation of this novel radiotracer with first-in-man administration, and here is the clinical trial number if you want to have more details about our design. And if you want to go into more details about all the programs that we can perform on Pharmimage, you can access to the website. In French or in English, both are available now, and to Oncodesign website as well. Next week we will be present at the ASTM meeting. There will be one poster on the IMAkinib project, showing the results that I just present to you. And there are some other posters that will be presented on other research programs of Oncodesign, and we will also be at a conference presenting a talk on innovative and predictive models against breast cancer. Thank you all for attending, and I'm available for any questions that you may have.
Okay, Cyril, thank you very much for this presentation. I hope that everybody appreciated this webinar, and I apologize again for the technical problem at the beginning of the presentation. Cyril, there are a couple of questions that came up during the presentation, and I just want to use the 5 next minutes to address some of them.
All right.
So the first question is, can you develop a new radiotracer in the context of a new pharmaceutical therapeutic program?
Yes, I think now, based on our experience, we have developed a full capacity from chemistry to clinics to really help our partners to develop their radiotracer. And I think this is a strength of Oncodesign and Pharmimage to be able to really put together all the expertise with the chemist, the preclinical pharmacologist, and the clinician are on the same table. And everything is available here on our site in Dijon.
Okay, thank you, Cyril, for this answer. There is one other question that I would like to raise here. How do you evaluate the in vitro slow binding release and correlate with the uptake of the radiotracer that you observe after 3 hours in vivo?
Yeah, this is a good question. At the beginning of the program, we look with Biacor for the time release, and we were surprised that actually the time release was quite fast, and there was no slow binding release. But in vivo, the compound is still present in the tumor after 3 hours. So we do not have a clear explanation yet, but it seems that the pharmacological behavior of the compounds within the cells or within the tumor is quite different than in vitro, and the compounds remain bound to EGFR for a longer time that we could expect from the data in vitro. So we don't know yet if it's a specific properties of EGFR compounds 4436, or this is something that we could extend more generally to the Nanocyclix kinase inhibitors, which actually we hope and it will be very interesting for a follow-up program.
Okay. Just before getting to the last question, I would like to apologize to anyone whose questions could not be answered, and we will get back to you as soon as possible. So, Cyril, the last question. How do you explain the improved binding to the double EGFR mutants compared to gefitinib?
I think the Nanocyclix compounds are very specific molecules, and with the linker, we are driving a 3D shape that allow us to go very deeply in the pocket, ATP pocket of the kinase, and we are not affected with the gatekeeper mutation as other TKI might be. This is something that is under evaluation, but I think the nanocyclic compounds behave differently than a regular TKI, an open form TKI.
Okay, thank you. So I want to thank everyone for attending this webinar today, and I wish everyone an excellent day or evening. Bye-bye.
Bye, thank you.